Okay. Okay. So the enzyme virtual lab that I'm virtually sharing, just shared with you, is the following. This one here, share this one. All right, so are you girls on that virtual lab? Looks like this. Awesome. So in this lab, this is a virtual lab setup. So what it means, right now you say lactase enzyme activity with data analysis. So if I'm gonna pin myself so that you guys can see me drawing on the board. All right. Okay, is it visible or is it, oh. When you guys are looking on the writing, is it flipped? Is it inverted?
We can't really see it actually. It's a little like
It's not flipped, but you guys can't see.
it's not flipped. It's not flipped, but we can't see it. Like the writing's really.
Right.
Maybe it was like orange or something.
Can you guys
Oh
see
yeah,
it?
okay, that's better. Yeah, we could see it now.
Okay, white on green is better. So lactose is a disaccharide. It is a sugar found in milk. Okay. Now, if you remember, it is composed of two monosaccharides. One of them is lactose. The other one is glucose. And this, okay, so lactose is disaccharide made of lactose and glucose. So usually when you're digesting milk, okay, usually when you're drinking milk and you're breaking it down and to digest it, your body will have an enzyme to break down the lactose to have these two simple sugars. The enzyme that is involved in doing that will be called lactase. Okay, the enzyme involved will be lactase. And this is one of the reasons why some people, quick show of hand, it's blurry and light. Better? Now, quick show of hand, anyone here is lactose intolerant? You're lactose intolerant, okay? Anyone else virtually? Better now, lactose intolerant, okay? Now, usually when you are lactose intolerant, so Sabrina, do you, what happened if you want a slice of pizza? What?
I eat it.
Do you take a pill or something like that after? So you just have cramps after, right? Okay, you suffer for it. Do you drink?
I drink milk. I drink almond milk.
You drink almond milk. So you try to drink milk that doesn't have.
Yeah.
Okay, lactose. Have you tried the lactose-free milk before?
Yeah.
Okay. How does it taste like?
I haven't done milk, but I've done the cream, and the cream is.
Okay.
Yeah. I don't know, like it's sweeter.
Yes, it is sweeter. Okay. Now, so Fusa said you should take lactate. Now, the thing is this. If you're lactose intolerant, your body is not creating this enzyme itself. So that's why you will have you'll have cramps because your body cannot break this thing down. Where you cannot break down the lactose. So if you take the pill lactate, the pill in itself will contain the lactase. Okay? The lactase, can you guys see up there? Yeah. in lactate. If you are drinking lactose-free milk, now lactose-free milk, what they do is they'll take regular milk and they will run the milk through a column, and in this column there will be beads that contains lactase. So when the milk flow through that column, the lactose in the milk will touch the lactase, and the lactase will break down the lactose. That is the reason why in that lactose-free milk that you get afterwards, it's sweeter because you have a lot more simple sugar now. Okay? So now, it's a very common experiment to do and to learn about enzyme because it's actually a pretty easy experiment to do. It's pretty safe. And I used to get my students to do that in other schools. Now, so if you look at the screen now, so if you look at this other screen, right now what you have here on the screen, right now here, this is a tube. That's a concentration of glucose, okay? Supposedly we have milk in there. Supposedly we have milk in there. So we can control how much lactose we have in here. You can have a lot of lactose. You can have very little lactose. You can control the temperature of the whole setup. You can control the pH, okay? So what happened is, so what you do, I do want you to try this out later on yourself. So if I switch off this button here so that there's no lactase added to this tube, what's going to happen? And I press the run. As time goes on, there's no lactose being produced, simply because if you don't add lactase in there, that lactose will stay there. And what the graph is doing is detecting the level of glucose. Okay, there are a couple of ways you can do it. You can use a glucose strip to do it, or you can use a sensor to record that too. There are a couple of ways to do it. Okay, any questions so far? People online. Okay, now if I reset this, now if I press the, I allow to have lactase in there. Initial lactose is 500 ml, temperature is 25, pH is 7. I press run. What you notice is this, you have this graph here going on. So you can see as time goes on, you have more and more glucose, right? As time goes on, there will be more and more glucose because the lactase was breaking down the lactose in there. You guys good so far? So that's what this initial graph is. Let me take a screenshot. Try to remember how to do this now. Nope. Oh, hey. Which folder is it? Okay. So now what I want you all to do is take a number of minutes, take about 5-10 minutes. What I want you to do is this. I want you to play around with the temperature. And I want you to play around with the pH. I want you to see how does it affect the lactose production. Because if it is faster, then this curve will be steeper. If it is slower, this line will be not as steep, it will be more flat. Okay, correct? Because faster reaction will be a lot of glucose, slower reaction take a long time before it reach. So play around with that first.
I have a quick question. Is galactose also a glucose or?
No, galactose is a disaccharide. It is made up of two simple sugar, galactose and glucose, connected together.
Okay, thank you.
Okay. So play around with it. I want you to vary the temperature, the pH, and I want you to vary the lactose. And I want you to record and see how is the rate of glucose production. Okay? So take 10 minutes to do that. By the way, make sure you press reset every time you did something different, okay? And I would like you to record it too. So either just quickly scribble down the graph or take a screenshot. So do that. It's going to do the walk down. What did you change? Oh, you make it more acidic. Okay. Over here. Increase the temperature. Now, maybe try to control one thing at a time, Sabrina. So change one thing at a time. So every time you change something, you record what it is, reset, change one thing at a time. That's a good thing, okay?
Why does it sometimes just be flat?
We'll talk about that. Okay, so yeah, make sure you reset every time you do something and then change only one thing at a time. This is what we called you only have one independent variable at a time.
So I have a quick question.
Sure, what's up?
I was looking back at the wanted poster, actually. This is a bit off topic.
Okay.
But after I handed in the wanted poster on Word to you, it seems that the formatting was a little messed up. So do you want me to send you a screenshot of my actual poster or try resending it?
That's fine. Why don't you send me the PDF? Usually if you create a PDF,
Yeah, I can
PDF
do that right now.
doesn't change format, okay?
Okay, I'll do that. Thank you.
Okay. So you can actually move these. Okay. Oh, keep on forgetting to use the white chalk rather than different color chalk. Okay, so let's talk about this. I think it's been about 5 minutes or a little bit more. Do you guys need more time to test out the different things? Sabrina shaking her head. Do you need more time? No? Anyone need more time? So in this case, in terms of temperature change, if you don't change any other things, you keep things as is, but you only change the temperature. Now I'm looking at this here. So if I'm comparing what I have before, this is, if it's 25 degrees, it looks like this. How would you say if I have the temperature, change it to lower temperature? Anyone tried 0? Anyone tried 10 degrees? What do you got? What happened?
Flat, almost.
Almost flat. Okay. So the graph will look almost flat, like this, right? This one, I'm just going to quickly draw. And what happens if it is higher temperature?
Also flat.
Also flat. But have anyone tried, like, let's just say 30 degrees? Like a little bit higher, but not super high.
It's also lower than that.
It's still lower than this. Okay? So even 30 degrees, it's still lower. I'm going to use a different color. So this one, this line is 25. 30 degrees will be lower. Right? Am I correct?
Wait, actually, it's like
30 to 35 was
a
good.
little higher at the beginning, but the end point is the same.
Endpoint is the same. Okay, so does it mean it gets to this level faster or lower? So if you compare the 30 degrees with the 25 degrees, is it like this or is
Yeah.
it like this?
No, no.
This one? Okay. Let me So 25 is this one. You're saying that 30 degrees will be sharper increase, but getting to the same level. Am I correct? Is that what you girls got too?
We got, or I did fly so much.
Is that what you guys got?
Yeah.
Okay, someone said 45-ish seems optimal. So it's a steeper. So this will be 45. Okay. Maybe you guys can't see that, what I have on the board. So 35. Okay, so 35 degrees will rise faster, but still get to the same level. Okay? Now, that's your virtual experiment data. But what does it mean? Okay? So now I'm just going to answer the question in order that was asked. So in this case, when the graph is flat, what does it mean? What do you think it means? If you look at the reaction up there, now I can't shift the board so it's up there. If the graph is flat, remember the x-axis is glucose concentration. Oh, x-axis is time, sorry. X-axis is time, y-axis is glucose concentration. What does it mean then? Yes, there's no glucose being produced. Why would that be the case? It's too cold, or what happens if it's extreme temperatures? So if it is too hot, if it is the temperature is extreme temperature. So at this point, let's see if there's another color that looks different for you guys on that. Okay, let me know if yellow on green is better. Can you guys see what I'm writing? Okay, so in this one, when the temperature is too high, what this means is your enzyme is denatured. Okay, it's too hot. The quaternary structure, the tertiary structure has lost its shape. In a sense, your enzyme's cooked. Seriously, 100 degrees, it's cooked. Good so far? Now, when it's too cold and you don't have the same level of activity, remember, the reason why chemical reaction happens, what?
I guess it's like there's no reaction, like energy being put in.
Perfect. So in order for chemical reaction to happen, I'm just recapping what you said. You need to have particles colliding with enough energy. If it's too cold, these particles don't have enough kinetic energy to provide the activation energy required. Okay? So here, particles too slow. Okay? Low kinetic energy. Ke is kinetic energy. Okay? Low kinetic energy. Now for 25, if you're comparing like in the middle, 25 and 35 degrees, why is 35, why would the curve look like this? What does it mean when 35, it gets up here faster and then go and then plateau? Yes?
I guess there's more energy for it to reach its maximum glucose production.
Okay. So in this case, what it means at 35 degrees, it reaches that level faster than 25 degrees. Means A, this will be the optimum temperature. This is the optimum temperature. This is the optimum temperature. 35 will be optimum temperature because it gets the rate of increase. If you remember this graph is steeper. It gets to the certain level faster than 25 degrees. The reason why 25 degrees is lower, it's either particles not enough energy or the structure of the enzyme is slightly warped, but not too much. So, from what I was told, 35 reaches this plateau faster than 25. So 35 would be the optimum temperature. For the enzyme, that's a temperature that the enzyme works best. 25 degrees, because it's lower than optimum temperature. So what it means is that the particles don't have as much energy, or if it is 45 degrees and it's slower, that just means that some of the enzyme starts to become denatured. Questions? Any questions? Now, my quick question for you is this. Why do you think both temperature, 35 and 25 degrees, reach the same level? Why is that? Yes.
Something about like the energy, like or it kind of like runs out and it like kind of just plateaus. Like, there's a high concentration of that, and then they kind of just like die off.
Can you rephrase what you're trying to say? No? I think you kind of get the idea of it. What happened is this. Remember, y-axis is a glucose concentration. And since here you only changed the temperature change, you didn't change the initial amount of lactose in there, whether it's 35 degrees or 25 degrees, you're going to get the same amount of glucose anyways, because both of them, you have the same amount of lactose, you're going to have the same amount of glucose. It's just which one get that level faster. So what do you think you have to do if you want this to be higher? What do you think you have to do if you want this plateau to become higher?
Yeah.
Not quite. I want this plateau to be higher. Remember, the reason why both temperature will have the same plateau is because they both start with 500 milligram per deciliter of lactose. If I want this to be higher, what do I need to do? More lactose to start with. Okay? The more lactose you start with, then this will be higher, assuming that temperature stays the same. Good? Let me write that down. Plateau, because same initial lactose concentration. I don't know whether you guys can see that. Yes. What? Well, this is I'm kind of teaching you guys why your graph look like what. Okay. If it's not on this test, maybe not next one, but the one after. Okay. Because the one after is called metabolism, and metabolism is enzyme controlled. We'll kind of revisit this a little bit more. Okay, same plateau because the same initial lactose concentration. Increase lactose concentration if you want to raise the plateau. Sorry for the somewhat messy handwriting with this. Okay, what I wrote there, just in case if you can't see my handwriting or the board, same plateau because same initial lactose concentration. If you want to raise the plateau, you need to increase lactose concentration if you want to raise the plateau. Close bracket. Good. And because I do want to teach it in a way that helps you understand why the graph look like such. So if you look at this, is it our room or the other? The other class, okay. So if you look at this chart, you would see this graph in the notes as well as in the textbook about enzyme activity, temperature, enzyme activity, what you will notice is that usually the shape of the graph look like this. The shape of the graph look like this when it's enzyme activity versus temperature. The peak here, that will be the optimum temperature. This will be optimal temperature. If that's when the enzyme activity is the highest. If you decrease the temperature from here, enzyme activity will slow down. Enzyme activity will slow down because this part, it will slow down because particles don't have enough kinetic energy, KE, kinetic energy, If it is higher than the optimum temperature, enzyme activity will slow down because the enzyme is starting to get denatured. So the reaction would not take place. So what I wrote here is enzyme starts to become denatured and can't catalyze the reaction. That's what happens when it's higher than optimum temperature. This is what happens when it's lower than optimum temperature. Write it down. Are the questions.
It says can't catalyze the.
Reaction, RTM, reaction. You guys okay? Questions?
So you're just saying like it can't break down the or like break down the reaction.
This part here?
Yeah.
Yeah, I cannot. Remember, enzyme is a catalyst. So basically at this point, the catalyst is broken. It's denatured.
Do you mind if I take a picture?
No, by all means. I was going to take a picture too.
Because the plastic counts. Thank you.
You're welcome. Actually, if you can post those pictures, can you post the pictures onto the Google Classroom? Okay.
I'll try.
You try? Okay. Other questions? I'm going to take a picture. So, all right. Are we good? Okay. So, any questions, virtual people? Once, twice. No. Okay. Let's talk about pH changes. Yeah, understanding how enzyme works is like one of those really important concepts in biology. So. Because if you don't have enzymes, we're probably not going to be alive. So let's talk about pH changes. So if you have pH of 1, you have pH of 7, if you have pH of, let's just say, higher, like 14. Okay. Again, looking at the graph, if it's pH 7, the graph looks something like this. What happens if it's pH of 1 or a really low pH? What did you guys have?
I think it was lower, but I'm not sure.
Lower or flat?
Yeah, like
Flat? Okay.
after I went under 2.8, it was like
Under 2.8. So if it's 2.8, it will still go?
it was like 0 point something, 1 point something.
Okay. So you're saying 2.8 is a pH, right?
Yeah, it was like low.
Okay, so a pH 2.8 is still low, but then here it's just flat. Okay, fair enough. How about high pHs?
Same thing after 11.
Same thing after 11. Okay. By the way, do you guys concur? Do other people have the same thing?
Like if you click on it, it's just like 1.8. I don't see like that.
What?
It looks flat. So if you click it, you click it, like it'll tell you like it's not 0.
Okay, good. Yeah, one really nice thing about this this particular virtual lab is that the guy who designed it built in certain random variables.
Yeah.
So even if you use the same number 3-4 times, you will get slightly different data, just like if you're doing it in real life lab. Okay, so that's your pH changes. That's the glucose level as time goes on. What does it mean then? Again, that's your experimental data. What does it mean? Which pH level works the best? pH 7 works the best. That is your optimal pH. That's your optimal pH. Okay. Okay, so you have between 5.5 to 7, that's the best. Okay. So a lot of people seem to get 7. Okay. So we'll just say 7 is the optimal pH for this particular one. Now, oh wait, green on green is horrible. So any pH that's higher or lower, you will see that the glucose concentration production will be low. Why is that? When you're changing the pH, what is being affected? True, but how is acidity and basicity, depends on the word. Yeah, different country have the slightly different textbook and slightly different term. Some will be alkalinity. How would that be different? What would those things affect our reaction? pH does not affect kinetic energy.
Would it affect?
Okay.
Would it affect the hydrogen ions?
affect the hydrogen ion, yes. So lower pH will have more hydrogen ion, higher pH will have less. But how would the higher or lower pH affect our reaction?
Would it affect the shape of the enzymes?
Yes, again, perfect. So higher or lower pHs will affect the shape of the enzyme. Will affect the shape of the enzyme. Now, in this case, at extreme pHs, when it's like 1 or 14, why it's flat or almost flat, is because it's completely denatured. Okay. Okay. It affects, when you have higher or lower pH, it affects the shape of the enzyme, completely denatured at either extreme pH. Good. Are there questions? Now the same thing also, so if you look at a graph that talks about, pH, enzyme activity, what you have again, graph will look like this, split in the middle. At this point here, that's optimal pH. That's the pH where this enzyme works the best. Anything higher or lower, as you move away from the optimal pH. Now, okay, yes.
And the textbook, it has like trypsin and pepsin.
Yep, we'll talk about that.
Okay.
I want to stick with lactase for a moment. Okay. So when pH are increased, it will affect the shape of the enzyme. Now, which part of the enzyme is the most important part to catalyze a reaction? There's a spot where the substrate will go in. What's the name of that spot?
The active site.
Active site, thank you. Active site. That is kind of the most important part of the enzyme. So the shape of the active site, you're talking about it. So at higher or lower pH, at higher or lower pH, the shape of active site is changed. Now you're talking about like a little bit different, a few enzymes versus all of them are different. Yeah. At the extreme pH. So that's why the enzyme activity is lower. Does enzyme activity lowered? I don't know know whether you guys can see all of it or not. Sure. You guys have any questions? Now, to answer your question earlier, you're saying pepsin and what?
Trypsin.
Trypsin. How do you spell the trypsin part?
Trypsin.
Oh, trypsin. Okay, trypsin. Now, pepsin and trypsin are different enzymes. Pepsin is to break down actually proteins. Okay. So now, in terms of optimum pH and optimum temperature, optimum pH and optimum temperature differs between different enzymes. Okay, so some enzymes 27, 37 degrees will work the best. Some enzymes want to be higher temperature, some likes it lower temperature. It really depends on where does the enzyme usually perform the experiment, or where do that particular organism lives. Okay. For example, there is a type of bacteria that we found in the Yellowstone Park. Okay, in Yellowstone Park in the U.S., there's a hot spring where people found a particular type of bacteria. Because we found that bacteria in the hot spring, the optimum temperature of that bacteria is like 89 degrees Celsius. Okay, where everything would be denatured, that's the optimum temperature. If we ever get to PCR, that is the enzyme that we're talking about. Some enzymes, for example, pepsin. Pepsin is the one that is an enzyme that breaks down proteins in your stomach. So if you think about it, your stomach acid is roughly the pH of 1. So your pepsin's optimal pH would be a lot lower. It would be like 1. Good so far? Questions? No.
So if that is what pepsin is responsible for. So if pepsin, I think that's the chemical you said, pepsin?
Yeah, the enzyme, yes.
Okay, so that's the enzyme like responsible for breaking stuff down in your stomach.
Yeah, breaking
What
down.
what does the acid do? Hydrochloric acid, I think that's what you mentioned.
Yeah, so in your stomach, you have hydrochloric acid. That will kill off a lot of the bacteria in there, and it also helps in chemical digestion. But it's not only the hydrochloric acid that's doing the job, it's also other enzymes too.
Okay, thank you.
Good. Okay. Other questions? Now, I'm not going to go too long on this. we kind of touched on this topic just now. So if you have glucose here, you have initial lactose. What were the numbers? 500. Versus just initial lactose is 1,000. We kind of talked a little bit about this when we were saying if temperatures, how do we want to raise the plateau if temperatures are stay the same. So you will have a graph look like this. If temp and pH are constant. Okay. So if you start with more lactose, you have more pH. No, higher plateau, lower plateau. If you have more lactose, you have higher glucose at the end. That's just because how much reactant there is. So that is also one reason why on the textbook or in a lot of graphs, what you have when you have enzyme activity, and you have substrate concentration. Substrate concentration. Okay. If you have a graph that has enzyme activity versus substrate concentration, what you notice is at this part here, more substrate, more enzyme activity. Okay. There's more substrate, there's more enzyme activity, but at a certain point, okay, saturation point, so beyond that saturation point, it's going to stay plateau. Doesn't matter how much substrate you add, it's going to be the same level of activity because at this point, all the enzymes are being used. They're doing as fast as they can. Okay. Think of like today, when there's so much snow, doesn't matter how much more snow there will be, you only have a limited number of snow cloud going. They're all working as fast as they can. Oversaturating. Are we good?
Sir,
Yes?
can you please show the right side of the board better just because it's harder to read on that side?
Your right side, right? That side?
Other side. Other
Oh.
side. Yeah. Thank you.
You're welcome. What? Complete what? Yeah. You do some review with it. What? And normally, if it's not COVID, I'll take a lesson just to do the lab and take a lesson to just hash these things out because it's an important concept. Now, are there any questions about enzymes and stuff that you guys have questions on? That opens for everyone, online or not. That side or that side? This side? Better, Taylor? So do you guys want to give me, do you want me to give you guys class time to just read through your notes and do those review questions? Sabrina nodding, you're also nodding. Do you guys also want that? Okay. A lot of people also want that online. So I will, at this point, stop recording. And I will give you the time to look through your notes. Right now,